Hi everybody. I just wanted to clarify a few things from today's lecture. And today you passed in your chapter 14 reading guide, and that focused on transcription and translation, otherwise known as gene expression, the cornerstones of the central dogma. In the previous chapter we had discussed replication, which is DNA making a copy of itself, called replication, and that's, we know, is essential prior to any type of cell division. So when we're looking at transcription and translation, it's important for us to understand the similarities and differences that happen between prokaryotic organisms, bacteria, and how those processes happen in eukaryotic cells. The primary difference is that the prokaryotic cells do not have compartmentalization, where the eukaryotic cells do have compartmentalization. We see that there is a nuclear envelope that separates the DNA from the rest of the cell and other organelles. And in the prokaryotic cell, the DNA is free-floating. It's a circular piece of chromosome, and so for that reason, we see that transcription and translation happen all in the same space. And because of that, they can happen simultaneously. Whereas in a eukaryotic organism, because of the separation, we see that transcription only happens in the nucleus, and that's the DNA going from to a pre-mRNA, and then that RNA is processed to a final transcript. It is that final transcript that exits the nucleus through a nuclear pore and then finds its way to a ribosome in the cytoplasm, where translation can begin and a polypeptide can be formed. So I just wanted to review that with this overview here. So we see that transcription and translation in prokaryotes and eukaryotes, it is essentially the same process going from DNA to RNA to proteins, but we see that those processes can happen simultaneously in prokaryotes. There is no compartmentalization, there is no mRNA processing, and this allows for the processes to be very quick and efficient. In eukaryotes, we see that there is compartmentalization. The processes are separated, and mRNA is processed, and we'll talk a little bit about that after. So I do want to be sure that I clarify one piece of information. I'm going to kind of hide this here a little bit so you can focus on this. So once transcription begins, ribosomes can attach immediately. So transcription is happening, and as soon as that mRNA is being produced, a ribosome can attach to it, and translation can immediately begin. Along that transcript, along that mRNA, multiple ribosomes can attach, therefore forming a polyribosome. But it's even a more complex polymolecule, because you have the transcript being formed as well as translation happening. Whereas once in eukaryotes, once the final transcript is sent out into the cytoplasm, at that point a polyribosome can be formed. So the steps are sequenced out and spaced out physically and time-wise, but once the final transcript is created, once that arrives in the cytoplasm, multiple ribosomes can attach to it and form the polypeptide simultaneously. Both of these processes allow for a relatively quick response if there are changes in the environment or there are changes in the organism itself. But this process, with multiple transcripts being formed while multiple ribosomes are attaching to each of those transcripts, that mechanism allows for a far faster rate of these processes occurring, and therefore gives them a greater advantage in terms of reproduction and the ability to acclimate to an environment. So I wanted to talk a little bit about the process of transcription first. So we're going to really focus on transcription. And in all organisms, transcription happens in 3 phases. We have initiation, elongation, and termination. In all organisms, there is a promoter and a transcription unit. The transcription unit is the section of DNA that is being transcribed. So the DNA is unraveled, the RNA polymerase attaches to the promoter region, and then elongation is the process by which the RNA transcript is formed. It is formed just like DNA is formed in a 5' to 3' convention, where the RNA nucleotides are added onto the 3' end. Termination happens where there is a signal which causes the transcript to stop, and then it is allowed to be released from the DNA, and the RNA polymerase is released. So let's take a look. So a completed RNA transcript can be formed. This is ready to go in prokaryotes, and this is ready for processing in eukaryotes. So let's make sure. So if we look at this process in prokaryotes, so this, in eukaryotes. So this is what the initiation looks like in eukaryotes, and this is more complex than it is in prokaryotes. So in eukaryotic transcription, we have a promoter region which contains a sequence called the Tata box, which is essentially a signal within the promoter that signals the RNA polymerase and additional transcription factors to locate and find the starting point. So the Tata box identifies where the several transcription factors will bind to the DNA, and then that complex signals the RNA polymerase to then bind, and then the transcription initiation complex is complete, and transcription, or the process of making a piece of RNA using the DNA as its template, it resumes. And again, it's just like we've said before, it moves in a 5' to 3' direction, RNA nucleotides being added onto the 3' end. So a little bit about the mRNA when we talk about it in all organisms, but we're going to really focus on eukaryotic RNA. So I kind of wrote a lot of stuff here. So if we look at all mRNA is read as triplet codons. So we look at a piece of mRNA and we read it every 3 letters, and those are called codons. In prokaryotes, the mRNA is functional as soon as it's made, but in eukaryotes, processing occurs. That processing includes several steps. So when the original mRNA is formed, that will receive a 5' cap, which is a GTP, guanosine triphosphate cap. This acts to stabilize and prevents degradation as it is moving from the nucleus into the cytoplasm. It also receives a 3' end, or otherwise known as a poly-A tail. Again, this is for stability, so it stabilizes and helps navigate the final mRNA transcript out of the nucleus. One of the things I want you to make sure you understand is what is wobble. If you don't understand what that is, we'll talk about that. But a hint is that it helps us to understand why some mutations aren't always harmful, and that has something to do with the fact that there are 64 codons but only 20 amino acids. So we'll hold on to that, but I want you to think about that. And I also want to look at the, oh, here it is right here. So we have pre-mRNA. So this is the whole pre-mRNA. So so once the RNA transcript is formed, we get the 5' cap and the poly-A tail, and then the introns are cut out and the exons are spliced together. So the introns are popped out and those exons are pulled together, and that creates our coding segment. So we have our 5' cap, our poly-A tail, and our coding segment, and we have little bits of transcript there that are not read. They're not part of the coding segment. So that is, I want to talk a little bit about how special that is. So pre-mRNA in eukaryotes contains introns. Now, how do you remember introns and exons? Well, introns are non-coding molecules. They stay in the nucleus. Exons are coding segments of the RNA, and they exit the nucleus. So there are two things that happen. Exons can be shuffled, and there's also alternative RNA splicing. So splicing is when the introns are taken out and the exons are spliced or kind of annealed or glued together. This occurs with a complex called a spliceosome, and that spliceosome is a complex of proteins and short nucleotide sequences, or snRNPs, and both of these factor into facilitating evolution and evolutionary changes, because exon shuffling and alternative RNA splicing can produce different products. If those products are advantageous to the organism, then those transcripts are going to be brought forward in that generation of organisms. I think that's it for right now. I really just kind of wanted to review that. I also want you all to think about the signal mechanism and how there are proteins that are slated to move out of the cell, and there are proteins that are slated to stay in the cell. And we want to think about that for our next discussion. Okay, thanks.